So, a warm welcome to all the students to this platform. I think it's a wonderful opportunity for you to learn about all the science which is happening in the realm of space. Today, I will be focusing on a life in the solar system and linkages to exosolar planets. The outline of my talk, and let's see, is going to first identify what Earth life is like, can be defined based on what is present on Earth, what could be life on other planetary objects, what are the basic requirements of life, what are the molecules, and can those macromolecules be divided into basic elements? What were the origin of these basic elements in our universe? What is the geological history of the Earth? And how these basic elements actually came about to become these macromolecules, which is life for us. And whether these life which is present on Earth can survive in extreme habitats. What does habitat mean? Is an environment where you can, survive and grow. So, what are extreme habitats? Something's very hot. Sometimes it's very, very cold, very high pressure, low pressure, no nutrients. So, can microbes or life on Earth survive in these extreme habitats? And going on from there, are there extreme habitats present in our solar system? And from there onwards, we will focus more on what kind of habitability might be present on Venus. Is there a past life or present life biosignature on Mars? What are these icy worlds we are talking about? And exoplanets. Okay, we found them. How would we characterize the habitability of these exoplanets? So I know it's very vast. So please tighten up your seatbelts and we'll hope to cruise through this. So I think when we think about life, it's mainly as astronomers, I think we come from this mindset of, okay, humans will be there, maybe animals, maybe trees, but what we always forget is about these unseen microorganisms which are present. They are present on the palms of our hands, in the biofilm of our teeth. They are present in our stomach, gut. They are present pretty much everywhere. So over here on the left, we can see that this is a microbial community. I have colored them falsely with pink, green, yellow, and they are actually surviving on these electrodes. Electrodes are nothing but, carbon, which is actually poised or, has a potential of a certain value, which can mimic the environment reducing potential. So whatever the environment might look like, we can actually mimic that on the electrode. And here I am showing you a hydrothermal well. These are like little magma or hot exposure of water which is coming from the deep subsurface of the oceans. And over here you can see that beyond these microbes, you can see these tube worms. These are eukaryotes. These are little animals which are surviving without light. So how are they actually surviving so deep below the surface of the ocean with no sunlight and pretty much minimum nutrients? So let's go on by defining what life is written as by NASA. So NASA actually defines life as a self-sustainable chemical system capable of Darwinian evolution. So if we ever go to exoplanetary systems or even extraterrestrial systems, what kind of life will be there? They have to be self-sustainable, and they have to be Darwinian. They have to have evolution. So this is a very limiting definition. So will a presence of virus which cannot replicate without a host not considered life? Are they not living? As in, we know that they are not living, but they definitely have the potential to live as long as they are associated with a host. So as we move forward, we have to really understand that the canonical characteristic of life are inherently the capacity to adapt to a changing environment and whether they can interact with other possible living organisms in the environment. So moreover, the definition of this life has evolved. We know that NASA just defined it very simplistically with saying that, okay, we have a self-containing, self-sustaining system, and we have Darwin evolution. Later in 2002, which is not many years back, Daniel Koshland said that it has to be programmable, it can improvise, that means it can actually interact with the environment, it has to be compartmentalized, actually separate from the environment somehow, it has to derive energy, it can regenerate, maybe it can actually replicate, it has to adapt, and slightly secluded. So these are the 7 pillars of life, which was defined by Koshland. And if we sort of combine these two in broad aspects, in 2007, the National Academy of Press actually released this nice little statement that any life has to have a way of deriving energy from its environment, should be able to maintain some bonds between elements, should have a liquid environment which can act as a solvent, and possibly have a molecular system that can support evolution. Evolution is not necessary, but this is all actually in the decreasing order of certainty. Like how often and how confidently can you define life? So in 2019, we have this very succinct definition of life, that a life is very far from equilibrium, self-maintaining chemical system capable of processing, transforming, accumulating information acquired from the environment. So what are the basic requirements of life? Of course, we have to derive energy. These energy can be derived from redox reactions. So actually you can take energy from lactate, transfer the electrons from lactate all the way to oxygen and giving you the highest amount of energy. You can also derive energy from sunlight, and we of course don't do that, but trees and many organisms do that, where they actually take in a very small amount of energy from red and infrared light and pump electrons from low energy environment to a high energy environment. And then when you drop back, you have an electron donor which accepts electrons. And this way you can derive energy. And the next important in the requirement of life is encapsulation. So you have to have this really nice molecular machinery encapsulated from the environment to permit maintenance of stability within a very chaotic environment. So while you have stability within the environment, the entropy of the overall system is increasing. Finally, and possibly when you learn, life is always learning from itself, you probably have the capability of self-replication using a genetic code, and it might be subjected to natural selection. So this is the central dogma of life, and you can see that to maintain all these requirements, you need macromolecules. So we have carbohydrates, which, for example, is cellulose, which is made up of carbon, oxygen, and we have chitin, which is the structural element of a lot of copepoids, and say, for example, shells of crustaceans like crabs and lobsters. They are made up of carbon, nitrogen, and oxygen. You have proteins where we have nitrogen and sometimes sulfur in form of cysteine. There is amine, we have a peptide bond. We have phospholipids, which actually form the outer membrane of our microbial cell. You have these nucleotides and nucleic acid like ATP, ADP, and DNA and RNAs. And what I'm trying to tell you over here is that these are nothing but basic elements, which is, for example, carbon, hydrogen, oxygen, nitrogen, sulfur, which is present in cysteine, and phosphorus, which are bound very intricately with covalent bonds, for example, sometimes hydrogen bonds within the nucleic acid, ionic bonds in proteins to make it more electrophilic or electrophobic. So all these are really important necessities of actually defining life. And here, so basic elements of life, I would like to, and in literature has been divided into 6 different elements, which is carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. And you can see how our macromolecules have incorporated them in their system. And if you look at a bacterial cell, which is the smallest cell which we can see, majority of this, which is gray in color, is carbon. And when you zoom in, you have oxygen, which of course we know that the CHO, which is is really important in all our macromolecules, and then hydrogen, then we have phosphorus and nitrogen, sulfur, and then these smaller elements which I call minerals. So metal ions are really important. They catalyze metabolic reactions, they stabilize the protein, they form gradients across the membrane, and they mediate signal transduction. And what I am very interested in is forming this, aiding in electron transfer processes. But where did we get all these elements from? So in the cosmic history of the universe, of course, we know that, as in this is an active area of study, but as of now, what I understand is that Big Bang fusion actually brought us hydrogen, and then we have helium, where we have contributions from Big Bang fusion, and we have exploding massive stars which can produce helium, and we can also have dying glowing low mass stars which can produce helium. And similarly, lithium, beryllium, boron actually come from cosmic ray fission. So it's all there. This is called stellar nucleosynthesis, and it was already present or is being formed continuously in our universe. So what is important to see is that these stable elements, when you rank in order of percentage of mass, so in the cosmic universe, of course, we know hydrogen is most present. I just wanted to know whether my laser pointer is visible. If not, oh my God, it's visible now. I am so sorry. Okay, so hydrogen is most abundant, followed by helium, oxygen, carbon, and then we have other elements which follow us. In the Earth's crust and Earth's ocean and the atmosphere, we again have a lot of oxygen present. We have hydrogen, we have a lot of argon and noble gases present in the atmosphere. And if we compare that with the humans have very well actually adapted, or even microbes have adapted to take up a lot of carbon in their system and oxygen in their system. So there's more oxygen in the system, and there's a lot of carbon in their system. So how did biology was able to grasp all these basic elements from Earth, which was present, which came from the cosmic expansion of the universe? So we know that the formation of Earth happened about 4.5 billion years ago, and then the formation of Moon probably happened 4.527 billion years ago. And there were these late heavy bombardments, which we probably think is because of meteoroids and asteroids hitting the surface of the Earth. They probably brought some carbonaceous material to help flourish life on Earth. These are all hypotheses, and it can also come from within the Earth. So this is all an active learning and research happening in the science right now. So again, we have, whatever the reasons are, first life, the biosignature of the first life was observed about 4.0 billion years ago, and the earliest start of photosynthesis. So photosynthesis is actually using sunlight to form oxygen, right? That happened 3.2 billion years ago. So what is happening here? When the first life was actually around 4.0 billion years ago versus the first sign of oxygen, what was happening in between these two regions? So I really was perplexed that when we started as planet, a lot of our seawater which was present on Earth was iron rich, and we had many more other chemicals which are present on the surface of the Earth. And as the metabolisms of microbial communities evolved, we had more and more oxygen present in the Earth. So here you can see that 4.0 billion years ago, we had a lot of iron in the sea, and as the microorganisms evolved and tried to learn how to make oxygen using anoxygenic photosynthesis, we got a little blip of increase of oxygen in the atmosphere. So on the top panel here, you can see how much oxygen was present on the Earth and how much minerals were present in the sea. And we can see the first blip of oxygen actually came from first organisms which are capable of probably respiring oxygen. So we had some oxygen producing, but there were some organisms which were also respiring on oxygen. So there was first blip happening, and then we had more and more oxygen accumulating on the surface of the Earth, and our oceanic system, the oceanic subsurface also was now rich in oxygen. So how do we understand that where these biomolecules, which were carbon, hydrogen, nitrogen, oxygen, and phosphorus and sulfur, how did they actually come about to form these nice little macromolecules for sustaining life? So there is, of course, this really interesting project which Miller and Urey did, where they have this nice borosilicate chamber in which they assembled an atmosphere which mimicked the environment of early Earth, and they were able to make a lot of amino acid, which was probably present in a meteorite also. And so they were able to see that. But what has now come into the picture is that could the borosilicate glass present in Miller-Urey experiment have an effect on what came about in the discharge experiment? So it really depends upon which researcher you're talking to. There are two ways of thinking about how life originated on Earth, whether it was geochemistry, everything happening in the atmosphere, or whether it was biology down approach. So whether Darwinian evolution was more important, whether you needed the building blocks of energy first, or whether you had to make something to actually catalyze all these reactions happening in a microbial cell, or was there no point of genetic or metabolism without compartmentalization? So what is most important? Whether compartmentalization is important, the genome is important, the proteins are important, or the metabolism? So it really depends upon who you talk to. They will tell you that RNA might have come first, or whether the metabolism might have come first, or the protein world, or the people who believe that encapsulation happened first. And it really doesn't matter what really happened first. It could have been happening together. But what really appeals to me is that every life is nothing but actually transfer of electrons, right? So when you transfer electrons from a protein molecule to another protein molecule is when you generate ATP in a biological cell. So coupling of electron transfer reaction actually forms ATP, which is mediated by this proton electrochemical gradient. Where can you find this proton electrochemical gradient? Inherently present in the Earth system. You can look at it under the deep subsurface. So in the ocean benthic floors, there are these hydrothermal vents where you have these geochemical gradients present. So you have a very alkaline hydrothermal fluid versus acidic seawater during the early Earth system. And when you have this gradient across a complex structure of metal and minerals, you can form initial macromolecules. So this person, Nick Lane, I would really suggest you look at his YouTube lectures. It's really interesting. You can actually make macromolecules, very similar, simple formate and glucose macromolecules by just combining carbon dioxide and hydrogen across a geochemical gradient. And this happens a lot in these hydrothermal vents. So the first image I showed you of tube worms present in the deep subsurface is happening without sunlight. All this energy is being utilized, and it comes from organisms which are living without sunlight, and they are living using proton electrochemical gradients. So geochemistry of these hydrothermal vents is giving rise to biochemistry. We could have had first protocells being formed, and then these proto-metabolism, which is the initial form of metabolism. And then these proton metabolism led to some RNA polymers, which is the RNA world. And we have these polypeptides being formed. And finally, we have a molecular machinery, which is encompassing within these microbial cell. And then our first LUCA, which is the least common ancestor, least universal common ancestor. So I think what I would like to say is that when you look at the thermal fluid versus seawater, the organisms are literally surviving on hydrogen and carbon dioxide, and it's a very reducing environment, which was the environment of early Earth. So I hope I have convinced you that probably hydrothermal vents were where life originated on Earth, and those kinds of environments still exist on Earth. So it is very well said in the marine community that humans and humankind know more about space than they know about the deep subsurface of the Earth. So I think it is, and if you want to understand life and how life might originate on other extraterrestrial planetary bodies, we must look deep inside the Earth subsurface. Okay, so again, going forward, this slide just tells you that microbes were the first organisms which actually came about, say, for example, 4,000 million years ago, and they're still surviving. They're still surviving through all these mass extinction, and probably will survive through climate change. So we need to understand how microbes have evolved and actually brought about life on the Earth. So again, moving forward, Earth is a planet of extremes. While on the surface, we don't see it because we have this really nice atmosphere which is protecting us, and we are not living in the deep ocean where the pressure is so high. So the pressures in the Earth can reach up to, say, 1,000 megapascals, and we have varied environments of pH, we have varied environments of temperature, and a lot of salinity. So how much salt is present? Of course, seawater is more saline versus fresh water. But humans can, and again, I'm not saying that we have humans living in this plot here. So humans can survive at 0.1 megapascal. Our blood is about 6.7 pH, and the temperature, of course, room temperature 28, 30 degrees Celsius, and we drink water, which is about 4 NaCl, right? But Earth life, which is the microbial life, unseen microbial life, has more extremes present. So they have been shown to survive in very low pressures, very high pressures. Of course, the hydrothermal vents, they are very high pressure environments. They have survived in very alkaline environments and very acidic environments. They have been able to survive at very low temperature and extremely hot temperature, for example, in Yellowstone National Park, and hot sulfur geysers of Ladakh. And we have very huge salinity also present where microbes can survive. And these kind of diverse extreme environments are also present inside our universe, right? So we have Venus, which we do not know much about the salinity, and we have Earth, we have Mars, where salinity has been predicted. We have all these moons of Saturn and Jupiter, where we have characterized what the pressure looks like and what the pH looks like. So can there be life in these planetary bodies in the solar system? Again, so we have to focus on the atmosphere and how the atmosphere of the solar system differs from the Earth. So they are all very different, of course, you would have seen. But let's focus on the Venus first. So the Venus has a lot of carbon dioxide. We have very little nitrogen compared to us, and there is a lot of sulfuric acid. Sulfuric acid technically is considered toxic for life, but there are microorganisms which can survive in acidic environment. So could there be life on Venus atmosphere? So if you look at the atmosphere and how the atmosphere changes, how the temperature and the pressure of the atmosphere above the Venus surface changes, there is a really nice sweet spot. So right above 60 kilometers, you have this right above this 60 kilometers, you have a temperature range which is very ambient for us, and a temperature range which is ambient for microbes also. So could there be any life which is living in these atmosphere in form of bioaerosols? So I know there has been much debate on the detection of phosphine in the Venus atmosphere. On phosphine as a biosignature is again debatable, but just to put it out there, phosphine can actually come from mineral phosphate by phosphide production, and that can be reduced by anaerobic metabolism to actually produce phosphine in the system. So there are microbes that can do that for you, and that is why Sarah Seeger, who was the main author on this paper, observed phosphine in the atmosphere of Venus from two different telescopes, ALMA and JCMT, and tried to inform us that there could be presence of phosphine in this environment, and it is important for us to explore Venus more further. So here again, I'm showing that could there be some microbes living in these encapsulated environment in the bioaerosols of the Venus atmosphere. Of course, NASA is never behind in exploring all these habitability questions in the solar system and beyond. So they are sending a lot of flybys and in situ analysis. They, in fact, have a balloon program where the outside is made up of titanium to save the entirety of the shuttle from sulfuric acid, and to be able to sample the atmosphere of the Venus. So now moving on to Earth, Mars, and how it is, how close it is to Earth in size, and also of course, all the planets were born together. We believe that it has a lot of mineral matter which could support life, because again, microbes are able to transfer electrons from a mineral to a mineral. So can microbes, or may have they been present in the early days, or early, not days, but early Mars. So again, we can see that while we have Earth here, we have oldest minerals and oldest rocks, we have the anoxygenic environment which converted into oxygenic environment, and we have these really nice pictures of the Earth environment looking into the past of the Earth, and where we can actually find what GOE, or the Great Oxidation Event, looks like. On the Mars, there are a lot of unanswered questions. What was the magnetic field like? Were there any hydrothermal activity? And were there any intense impact or bombardments? And could there be any signatures of life on the surface of the Mars? Moreover, we have discovered that Mars hosts a lot of hydrous minerals. So you have hydrous clays, you have hydrous sulfates and carbonate salts present on the surface of the Mars. So while the atmosphere is very thin and you have a lot of sun exposure, so surface might not be able to host any microbial life, or any form of life for that matter. Could there be life in the deep subsurface? For example, if we drill a core, or take, say, 5 meters or 5 centimeters of the core on the surface of the Mars, can we find signatures of current or past life present on Mars? So moving on to these icy worlds, and these are very far from Earth, and of course from Sun, and that's why some of these moons of Jupiter and Saturn host very high and deep ocean environments, which are possibly directly interacting with the rock or the crust at the center. So there are knowledge that we have these, of course, there are photographs being taken, that there are plumes which are coming out of the surface of Europa and Enceladus, both which are the icy moons of Jupiter and Saturn, respectively. And could there be any hydrothermal vent like feature which could be present at the deep subsurface? Again, reminding you, Earth subsurface is Earth ocean is not very deep compared to what we are looking at here. Here, the deepest we go down to is 100 kilometers. So what kind of instruments will be required to look into that deep water rock interaction? So NASA is sending Europa Clipper mission where they are going to incorporate some radar instruments to see how deep this the depth of rock and water profile could be, and whether they can map any gases coming out from these environments using mass spectrometry. So one hypothesis is that the olivine and water which could be present in the rocky core can produce methane, which is probably an abiotic source of methane. And once you have these methane can possibly come out in the form of plumes, because methane was detected on the surface of the Enceladus ocean. So the next question is, if we have methane, can there be any methane metabolism? So microbes of the early Earth have been shown to metabolize methane, so which is called methanotrophy. So it is important to figure out that whether we can have any microbes present on these environments. So NASA is again sending and identifying all these ocean worlds which are present within the solar system, characterizing these oceans, what kind of solvents may be present, what kind of rock ocean interface is present, and would there be any energy for life? So for example, we need to understand that if there is life, where are they deriving their energy from? What kind of environment is present? And finally, searching for life. Well, can you just take an image? Just an image will tell you whether there's microbe present or not. So this is a very this is an area of active research of how to identify potential biomarkers in extraterrestrial system. So moving on, we have, of course, these exoplanets. There are different kinds of exoplanets, and about 5,000 of them have been found, which are planets, like Earth, or gas giants, or maybe super Earth, or probably Neptune-like, which has a lot of water, warm water on its surface. They have been characterized in this exoplanet archive. We have to identify which one of them would be habitable. So, of course, there are various ways of identifying exoplanets. There's radio velocity transit, which was led by JWST, actually works on the principle that when a planet comes in front of the star, which it is associated with, the atmosphere or the emission spectra or the transmission spectra which is received on the Earth or by the telescope changes. So, and whether there is a duration to that change, and this sort of helps us identifying whether this is a planetary system or not. But to sort of understand or even speculate whether this exoplanet may be habitable, there are various questions and answers to be given, right? So whether it is a minor planetary system, does it have exomoons? Will there be dust storms on this planet? What kind of atmosphere there is? Whether there is any stellar effects. So what is the rotation like? What is the magnetic field like? Are there any cosmic rays? Because if there is any cosmic rays, and if there is no atmosphere, of course the surface will not have any life. If we look at the biology aspect of it, we have to study the atmosphere, which is the albedo. We have to understand what is the mass and the radius. We have to understand what are the temperature, composition, and weathering effects. Whether there is any UV shielding, and whether there is any kind of climate. So all these studies will actually inform us whether there's any habitability possible in these exoplanets. And very briefly, Sarah Seeger in 2013 sort of identified that we have, if you want to see whether they're Earth-like and if they're habitable, we can look into how the radius of the orbit looks like compared to the Earth versus how the mass of the star is relative to the Earth. So if we look at this habitable zone, which is blue, we can identify which of these exoplanets are possibly habitable for any search for life. 1 second. So with that with that taken into picture, of course, there is Kepler 432 system, which is pretty much the sun is about twice as big as the Earth sun, but the star is twice as big as the Earth sun, and then we have the distance about the same as Earth. So this is a very popular planetary system which is being studied in for exoplanet habitability. So again, coming to transmission spectroscopy and how the planets orbiting around the sun can help identify what kind of atmosphere which is present on that planet. So this is a work from JWST. Of course, you can see that the wavelength actually spans from IR all the way to close to UV, and you can see that a lot of little elements, biologically relevant elements, were identified in the spectrum. So NASA is sending again a large UV optical infrared instrument to identify various planetary system and their absorption transmission spectra, and to figure out whether there could be or what kind of atmosphere might be present in the planets, and whether it can be habitable or whether there can be any life in the system. But we need to take all these research with not a grain of salt, but with some understanding that when any research is reported, we need to understand it should tell us the status of life detection, whether you have detected some kind of biological activity. Can you rule out the contamination present in the spectra or otherwise? And can you demonstrate that there would be that biological production is the sole reason of that and of that detection, and whether there are any abiotic sources present? And then, do you have any independent signal coming from biology which can be detected? And I think we need to do this in a more systematic manner and understand that whether we can have a follow-up observation in time to see how the atmosphere changes in time to understand the effect of atmosphere. So here you can see, if we look at the same spectra for an early Earth versus what the modern Earth looks like, it is changing. So if we go back to the same point and try to understand how the spectra possibly has changed within a year or two, we might have more clues to possibility of life on that exoplanet. And with that, I would like to quickly summarize that yes, life is a self-sustaining chemical system capable of Darwinian evolution, and we will accept that. But we need more information, and we need to be more thorough about defining life. And life over here is based on what life we know as on Earth. Life could be something beyond on Earth. Carbon may not be the basic molecule which would form the backbone of macromolecules. It could be something else. Water may not be the solvent. It could be sulfuric acid, or maybe something more reactive, maybe an alkane. So we will have to think about all those questions. We need energy, we need a solvent, we need encapsulation with a constant way of exchanging information from the environment. We need these bonds, not just covalent, but every bonds. We need eventually some form of replication for life to thrive in that environment. Of course, macromolecules are carbohydrates, lipid, proteins, and nucleic acids, but they're all made up of these basic elements, which is carbon, hydrogen, nitrogen, oxygen, and phosphorus, which is called SNOPs in short. Basic elements were present before life evolved on Earth based on stellar nucleosynthesis. So, what life had to do was just bring them all together and make them relevant so that life could actually exist. And then the geological history of Earth is very dynamic in space as well as temporally. So it's a different field of study altogether. Mm, For me, the original building blocks of life can be defined by just how geochemistry of early Earth probably gave rise to first biochemistry, biological molecules, and how those biological molecules probably came together to be encapsulated in a more uh, microbial life kind of a format. And extreme habitats are present pretty much everywhere on Earth. The first microbe survived only on redox chemistry, which was present around in the environment, carbon dioxide and hydrogen. And it was very late in the timeline when they actually started producing oxygen. So there was a very big gap of microbes which are surviving on anoxic in an anoxic environment. There are extreme habitats which are present. Habitability on Venus can be in the sweet spot in the atmosphere. There is, of course, a lot of carbon dioxide rich water present on Mars. There could be some past images of life in the subsurface. In Enceladus, Europa, they have very thick ice shell, and we need to study water rock interactions on the seabed. And habitability of exoplanets, we have to look into many criterions. The need of the hour is to incorporate how to identify these life detection scales during our habitability studies. And with that, I will be taking questions.